Hello, and welcome to Everything I Need to Know About Survival I Learned from Microbes. I think that's what we're calling this event. Is that correct?
I think so. I think we need to apologize for that music. It sounded like I was playing Mike Tyson's Punch-Out and I needed to reinsert the cartridge on my Nintendo. Sorry, everybody.
Okay, I actually was thinking as we were listening to that, oh, I don't like this music, but thank you for sticking with us. At least we hope that we didn't scare you away with the music, but my name is Ann Merchant and I work for the National Academy of Sciences, and I did not pick that music.
My name's Rick Lovard. I was part of the process of picking that music, but it accidentally got played over itself twice. That was the issue. So there was a glitch. Apologies for the glitch.
Well, it can only get better from here. That's all we're going to say. And so welcome to today's event. I often start by saying a little bit about something about the National Academy of Sciences. Sometimes I try to pick things that directly relate to today's event. I think that this relates only to say that the National Academy of Sciences is host to the Schmidt, and I want to get this right, the Schmidt Awards for Excellence in Science Communications. And of course, a lot of what we do at the Science and Entertainment Exchange is science communications, and Nkrumah will be doing something that certainly is a wonderful example of science communications. And today they announced the first of the awards that come out of that program. And they are awards for science journalists and also for research scientists who are doing very creative things that essentially communicate to broad audiences around the public communication in STEM topics. And so I encourage you, I believe that Sachi has put in the chat the link to those awards. I encourage you to check them out, not only to see who the winners are, but to see the examples of the fine communication that they're doing, because I think that they're all worthy of a look. So I would love for you to do that. But of course, as I said, there's a lot of the work that the exchange does is around science communication to particular audiences, such as yourself. But Rick, if you would like to go ahead and talk a little bit about the exchange, that would be great.
That's right. Hi, I'm Rick Lovard. I'm the program director of the Science and Entertainment Exchange, which is a program of the National Academy of Sciences. The basic programmatic purpose is if you are a storyteller in a mass media format, a writer, producer, a director, a studio executive, you're making a TV show, a video game, a novel, a graphic novel, whatever it may be, a documentary film, you can call us and we will connect you with an expert who can answer your questions and put them in the context of your story. We've done this over 3,500 times since we opened our doors on many of your favorite TV shows and video games where grown-ups are playing make-believe. If you're interested in a recent example of our work, I'm going to go ahead and mention Paper Girls again, because I thought it was great. And also we're friends with one of the people who worked on it. So, you know, little inside baseball there. And if you are a STEM professional and you are interested in being connected to storytellers or plugging into our community, especially now that we're starting up our live events as well in Los Angeles, we're not going to stop these events. We're still going to be doing our online events, but we're also going to start doing live events in Los Angeles and probably eventually New York, probably, right? Yeah, okay. Then if you are a STEM professional and you're interested in the work we're doing, please do reach out to us. Sachi's going to put some contact info down below here. So you can find a recording of the show on the Exchange's website after the fact, usually posts around Friday, sometimes early the next week, usually by Friday. Or you can subscribe to the Science and Entertainment Exchange on YouTube to see these events and other videos like our Ask a Scientist series. You can sign up for our Collider newsletter, which goes out weekly. When the video for this event is available, everyone who RSVPs is going to get a link. I want to thank our sponsors today, Howard Hughes Medical Institute, without whose support we would not be able to do any events ever. And also we get major funding from the Alfred P. Sloan Foundation. And I want to also thank individual donors, all of you who donated to today's event. It goes right back into doing more of these events. So thank you very much for that. In a moment, you're going to hear from Nkrumah Grant. Anne will tell you more about him in a second, but he's going to speak. If at any time during his talk you have a question about things that he's talking about, please throw it down here in the Q&A. I know not everybody likes that we disable the chat. I'm sorry, we need it for the back end, but if you have questions, I will be dutifully making a list and I will get to as many as humanly possible. We're going to pop back on after Nkrumah finishes his talk, and you can keep asking questions during the Q&A. We also have a VIP Q&A that some of you, I think, have signed up for. If you did sign up for it, or you are a supporter level donor, then you should have gotten a link, or a retreat alumni. If you are in any of those categories, you did not get a link, please direct message Sachi right now. I'm sure she will appreciate that, but she will definitely get you a link. So my rabbit hole for this week is about how microbes in our gut may impact our mental health and lead to possible new therapies for depression. So I thought that was super interesting. And back to you, Anne.
It is super interesting, and I plan to follow the link to your rabbit hole. My rabbit hole is to the Carl Woese Institute for Genomic Biology at the University of Illinois, which is run by a friend of the exchange and a member of the National Academy of Sciences, Gene Robinson. And for those of you who do not know, Carl Woese in the 1970s, his research suggested that there was a third branch of life in the tree of life, and he sort of exploded our understanding of biology in that way, that there was a third kingdom of living organisms. So if you haven't heard of Carl Woese, I suggest you go look him up, look at the IGB. They're doing super interesting work, and you can learn a little bit about an iconoclast in science, because who doesn't like somebody who revolutionized science? But, you know, we have the great pleasure of meeting a lot of people who really push the boundaries of science. And in particular, Rich Lenski, who is a member of the National Academy of Sciences and is on the NAS Council, the Executive Council. We are required to report back to the council on the programs that are NAS programs, such as the Science and Entertainment Exchange. And so I was giving an update to the NAS Council and talking about the work of the exchange. And I'm always, you know, encouraging them to tell us about the work of interesting people that they know, so that we can reach out to them and feature them on our programs. And Rich had reached out to me immediately and said, you know, I know somebody that you need to know, because I think that he would be great for some of the programs that you're working on. And that's how I found out about Nkrumah. And I have to say, he sent me a link to Nkrumah's webpage. And Nkrumah, you're welcome to join us now. But I have to admit to you, I read your bio on your webpage, and I read what you do, and I thought, I don't actually understand this. I'm not sure I understand any of that. And so I had written to Rich, and he explained to me what you do. And I said, okay, well, not only does he sound like a really interesting guy, but now I understand a little bit more about his work. And that sounds super cool. And so that's why we have asked you to come and talk to us about your work, because in the spirit of Carl Woese and in the spirit of pushing the boundaries of science, it sounds like you're doing some really interesting things. And we know that our audience would be very interested in that. So thank you for being here today. And we're going to hand this over to you. And I know you have a slide deck, so we're going to get out of here and give you the whole Zoom screen to do what you do.
Don't forget to unmute. Oh, there you are. Good.
Got it. We're all set. Perfect. Let me just set this up and make sure that we're all set here. You all should be seeing my slides, correct? We good? I'm going to assume so. No one has yelled at me yet. So first, I would like to thank Rick and the National Academy of Sciences and the Science and Entertainment Exchange for inviting me to speak at this event. Today, I'm going to let you, take a dive into my mind and share with you all some of the thoughts that have kept me up at night for a very, very long time. But before I begin, I would like to make a nod to this background image, which is among one of the first images that was taken by the James Webb Telescope. It shows the region of the Karina Nebula, which is a region deep in space, where stars are born. And on it, I have superimposed a representation of a bacterial cell and DNA. And I know that Anne and Rick, they titled this talk, but if I were to give it a subtitle, it would be How I Knew I Was a Microbiologist and What Made Me Want to Become a Microbiologist. I think this image captures that moment. I came to this realization because it's the moment that I realized that life can exist in space. So today I'm not going to tell you too much or give you too much data, except in the context of what it is that I have done. I think that it's how it relates to the work in putting microbes in space, but more of a conceptual idea of what is required and what we can do to put microbes in space. But before I go there, I would say that microbes too often get a bad rap. I mean, currently we're living through 2 pandemics in the terms of, monkeypox and the coronavirus. And then in the news, we always hear things about these antibiotic resistant superbugs. And I would say that this bad rap is kind of indoctrinated into us from a very, very young age. So here I'm just showing a kid that's running from the proverbial germs, which might as well just be my mother, my father, and my grandparents when I was growing up telling me not to do anything because germs are all around. But I'm not saying that microbes themselves don't do horrible things, but let's look at the numbers itself. So there's an estimated 10 to the 11 to 10 to the 12 microbial species within the biosphere. That's a 1 with 11 and 12 zeros, right? And only about 1,400 of those cause disease. That's less than 1%. I mean, extremely small percent of microbes that we know about cause disease at all in humans. So for me, I believe that it's unfair to treat all microbes as villains or as germs, and that we should learn to embrace them. Because when we do, we will see that microbes are abundantly diverse. Now, I don't have any fungi here, but those could be included in this image as well. Microbes that come in different shapes, different sizes. They can live as free living cells or in communities where they barter internal and external goods between each other. Some fight, some enable each other, but microbes are among the most successful species on Earth. And I would say that indeed, we as humans, we can learn a lot from microbes about working together despite our individual differences. I would also say that when we look at any place in the world, they are found in every corner that we look. Of course, they are found on the inside and the outside of our bodies, where they outnumber our cells by an order of magnitude. We can find them in places like this place, where this is in Ethiopia. It's called the Dalal geothermal field, where we have acids that spew out from the earth and temperatures that get as hot as an autoclave. In contrast, we can find them deep in Arctic ice, where they produce antifreeze so that their cytoplasms don't freeze over. And if you think that radioactive materials are off limit, forget it. Microbes readily dine on this. And space is fair game as well. Some bacteria species, they are master hitchhikers. They find their way to space in jet fuel. Now, as a microbiologist, as a microbial and evolutionary biologist, I'm interested in the strategies microbes have evolved to live in these environments. And my belief is that understanding the genetic and physiological adaptations that govern their existence can inform conservation strategies in the face of changing selection pressures like climate change, and also inform design principles in the realm of bioengineering. So that's kind of like my main global interest, but I have other interests as well too. And one of those is that seeing that microbes have evolved to inhabit every corner of our biosphere, is it possible that they can inhabit other planetary bodies in our system? So indeed, other planets in our solar system like Mars and the atmosphere of Venus, or moons like Titan and Europa, harbor conditions that might support extant life. And that's fine. And also, how might that understanding of the physiological and genetic adaptations that allow microbes to survive here on Earth help aid in the discovery of life in these systems, or better yet, what I like to think about, provide insight for how to engineer microbes capable of living in them? And this is the kind of questions that a global field called astrobiology is concerned with. And this is something that I don't think that many people, or at least I remember when I was growing up and thinking about science, didn't know existed. So this is out there. So when people think about aliens, or I'll say microbial aliens, just know that there's a whole field devoted to thinking about these type of questions. They're not crazy. Now we can't go back in time and think about all of the places that microbes live, how they got there, and think about and know at least the genetic and physiological adaptation that allowed them to thrive in those environments. But what we can do is experimentally evolve modern organisms under controlled laboratory conditions and impose a selection pressure, which is what the field of experimental evolution is. Now, experimental evolution has been done with many organisms, but they're usually done with single-cell organisms such as yeast and bacteria. They can also be done with proteins and DNA sequences, and they can also be done computationally. And this is because these systems, they have rapid generation times or rapid turnover rates. They have simple genomes, or they're comprised of simple instructions that we can follow over time. And they're able to be preserved. Evolution is able to be preserved at intermediate stages. And what do I mean by that? In the case of the bacteria and single cell organisms, for example, we could freeze down samples over time. And in the case of a computational system, we could save data as we generate it on a computer. And experimental evolution is fairly simple. So here I'm just going to give a hypothetical example of what an experimental evolution experiment would look like. So here, for example, you can start with an ancestor and some condition that you're interested in studying or evolving or looking at the effects of adaptation in. So here I just have space in the background because that's what we're talking about today. And I have this very, very hardy organism called a water bear or tardigrade that can withstand high temperatures, high doses of radiation. It can dry out for years at a time. And then once water comes back around, it can revive itself and act like nothing ever happened. So let's just say I want to see how this organism evolves in space and what happens there. I would take the ancestor, or I would take a tardigrade or a population of tardigrades, and I will sequence that tardigrade, and I will put it in my environment. I'll give it the food, and I'll put it under space-like conditions, for example. And then over time, they will consume the nutrients or whatever that they're using to grow. And then I'll back dilute them out into new media, And I could repeat this until infinity, some amount of time, until I retire, or something of that nature. And then I could look at the ancestor and derived clones. Now, just because I could then go back and sequence the derived populations, which then provides information about the genetic changes required to live under the imposed selection pressure. In this case, I could say what happened to the genome that allowed this organism to survive and do better, or survive and thrive in this environment. Now, one such evolution experiment, and the longest running evolution experiment, long-term evolution experiment to date, was pioneered by Richard E. Lenski. And he used E. coli, our model system, our model bacteria, to do so. And he was interested in three kind of motivating questions. What are the dynamics of evolution, including the rate of change? How repeatable is evolution? And what more can we learn about the relationship between genotype and phenotype? So he took these 12 populations. They were started from a single common ancestor, and he put them in a simple, very, very simple media, and did the transfer regime as I showed you on the previous slide. Now, upon joining Rich's lab as a graduate student in 2014, I wanted to do something that would get me a little bit closer to thinking about life in space. And one thing that I was interested in, or that I realized, is that E. coli itself is a facultative anaerobe, which means that it's capable of growing in the presence or absence of oxygen. And I'm going to call this metabolic plasticity. Now, by virtue of how these populations are growing in Rich's long-term evolution experiment, these populations, there's no selection on anaerobic growth. The flasks are constantly being shaken. There's a lot of oxygen and influx of oxygen into that media. So what we say for the anaerobic metabolism, it is under what we call relaxed selection. So one question that I really wanted to know was whether or not metabolic plasticity was retained. And I wanted to know if I could understand this from a genomic and phenotypic basis of the trait. So to answer this question, there is some metagenomic sequence data, which is just a deep sequences taken from each one of these populations of every single cell, just about, that tracks all the mutations present in those flasks. And it was taken up through 60,000 generations of the experiment. I then also used a publicly available data set and I identified genes that were differentially expressed exclusively in an environment with oxygen or in an environment without oxygen. So we call this strict aerobic or strict anaerobic genes. And what I wanted to know is what is the mutation frequency in these genes? That was one thing that I wanted to know in doing this experiment. I had some naive hypotheses as well for what would I expect to happen, right? And this is just kind of like generally in the field. From the genomic perspective of the trait, I would expect there to be a higher abundance of mutations in anaerobic specific genes when compared to those in aerobic specific genes. And there's a reason for this. So genes are only, mutations from genes, they're typically deleterious, and they're only purged from a population typically when they're being expressed. So because these organisms are not in an anaerobic environment, these genes are not being expressed. So essentially they serve as buckets for mutations. There's no selection to remove those. And then from the phenotypic perspective, I expected there to be stunted performance of evolved lineages in an environment without oxygen when compared to the ancestor or earlier descendants. And that just makes sense. They haven't seen it. They haven't seen this environment. They're getting these mutations. They're degrading. You don't use it, you lose it. So here what I'm showing you is some data that I collected during that experiment. So what I'm just showing you here, and you don't need to worry about the axes, but each one of these panels, they show the mutations that occurred in each one of those 12 populations, and those are colored, right? And you can see that some of them have more than others, and those are populations that evolve what we call hypermutability. They evolve the capacity to just produce more mutations. Now, again, I expected because there's all these mutations in these populations that anaerobic specific genes should have more mutations. And that is exactly what I saw. So here I'm just showing you the number of mutations on the y-axis, and on the x-axis, I'm just showing you the environment. And as you can see, the anaerobic related genes, so genes that's required for growing in the absence of oxygen, have approximately 3 times more mutations. Now, again, given the fact that most mutations are deleterious, and if we look at these mutations as well, we see a lot of them, we do see a significant number of IS elements. So these are just genes that jump around in the genome, like big chunks. And when they integrate, typically, they should break function. So we see a lot of these, so we would expect to be stunted performance in an anaerobic environment. And actually, that's not the case. So here I'm just showing you the fitness is measured by taking the ancestor and evolved clones and mixing them up together and growing them up in the same environment, either in the presence of oxygen or without. And I'm looking to see who competes and who can get the glucose faster than the other. And in the aerobic environment shown here in black, so that's the environment with oxygen, we see an increase in fitness such that by 50,000 generations, the average population is on average about 55 to 60% more fit. In the anaerobic environment, unexpectedly, we also see fitness increase, though by 50,000 generations is only about 10%. But we do see that there's a significant fitness increase between 2 and 10,000 generations. So not only have these populations maintained the ability to grow in an anaerobic environment, they've also improved, despite not having seen this environment for 50,000 generations or roughly 30 years. So what this means, or what this tells me then, is that microbes can maintain essential metabolisms over long periods of time, even in the face of genetic perturbations. And when we think about this result in the context of space, where there's high doses of radiation and other chemical mutagens, understanding then the genetic architecture that leads to this robustness could prove critical to genetically engineering evolutionarily stable features into our microbes. Now, in addition to that, microbes do useful things. So they produce medicine. So a lot of our antibiotics, kanamycin, amoxicillin, they come from fungi, right? So that's one thing. We can also engineer them to produce things like insulin and vitamins. They also can produce natural products, plastic fuels or proteins for foods. I told you earlier about the bacterium that produces antifreeze to maintain their cytoplasmic fluidity. And also, at the end of the week or the end of the day, to each his own, they can also make beverages as well. Those fizzy drinks that we like that gets us a little woozy sometimes. Now, in my first postdoc at the University of Idaho, I had the advantage of working with a model organism called Methylobacterium Extortions. Now, this organism is a pink pigmented soil and aquatic bacterium that is readily isolated from leaves. And they're unique in their ability to use single carbon compounds, including methanol for growth. So just by comparison, we can use several sugars, but glucose is a 6 carbon compound and it's something that we use. Now, because they can use these single carbon compounds as feedstocks and their ability to strive under stressful conditions, so when they use these single carbon compounds, they have to go through toxic intermediates like formaldehyde. This makes Methylobacterium an attractive microbe for biotech. So while at the University of Idaho, we were concerned with using this organism to produce goods, particularly plastics. And I'll show you how we did that. So lignin is the second most abundant biopolymer on Earth next to cellulose, but it's highly recalcitrant or hard to break down. It has all of these aromatic groups. So while it has all these aromatic groups, so while we are interested in using lignin, we typically can't unless we input a lot of energy to it in the form of petrofuels. However, it is known that microbial communities can break this lignin down to smaller chemical units that are easier to use. So while at the University of Idaho, I simply asked a question with my the PI there, Christopher Marks, can we engineer microbes that are able to use these smaller units and convert them to useful products? So to do that, we engineered a novel pathway into Methylobacterium extortions that made them capable of using these aromatic compounds, particularly vanillic acid and protocatechuic acid here. And the idea was that if we can introduce these pathways, they'd be able to grow on these compounds, which is a plant waste, right? And later on down the line, as research progressed, we can actually cut those genes out and introduce some others. And the energy that they use to produce these compounds could then go and be fed into natural gas like butanol. So after we engineered these populations, I performed experimental evolution, and I evolved the bacteria to see if and how they improved. And this evolution experiment I performed for just 20 weeks time, which equated to about 120 generations. So I just want to say that there. So here I'm just showing you some of that data. On the y-axis, I'm showing you the doubling time. And on the x-axis, I'm just showing you each of the populations that I started, and I also had some replicates of each of those populations. So when we think about doubling time, if you're higher above, that means that you have a slower growth rate compared to other ones. And what we can see is that when compared to the, our evolved strains are in, I can't see on my side, our evolved strains are in green, our ancestors are in red, and I have a natural isolate from the wild that is able to use some of these compounds, basically serving as a control. And what we can see is that where the ancestors, they take, on average 10, 15 hours or so to grow on these carbon substrates, the evolved populations actually do so in half the time. And they approach the rate at which the natural isolates do it. And here's the same data. I'm just showing you the grand average or the grand mean of these populations. And you can see that much clearly, that the evolved populations on both substrates, protocatechuic acid and vanillic acid, they improve. They're 60% or so faster than the ancestor when grown on this novel carbon source. And this just means that microbes can learn to live with the introduced metabolic pathways over very, very short evolutionary periods of time. Again, this is 120 generations, and they've improved by 60% relative to the ancestor. And what else are they doing with this? So if we look at them microscopically, we can see, don't worry about these genotypes. We have a couple genotypes I looked at as well. We can see these small little granules inside of them, which means that they're producing plastic, the things that we want them to grow and want them to produce, right? So that's really, really cool there. Now, I told you 2 quick stories, and I introduced this topic as a means of putting life in space. And I will say that many species of Earth's biosphere are space ready, right? I'm just gonna give an example of one, or two of them. This is known as the Sunburst Lighting. It actually typically grows on rocks in sheltered environments. And interestingly, there was an experiment done with this organism where we sent it to the ISS, and it was actually on the outside of the International Space Station, and it remained viable after 18 months. So we could bring it back down to Earth and we can see and grow it up, and it was able to remain, happy, like, it never was up there. Dinococcus radiodurans is another organism which is dubbed as the world's toughest bacterium. It can withstand about 5,000 grays of acute radiation with no loss in viability at all, right? Or 15,000, even higher, with 37% viability. By comparison, only 5 grays are lethal to humans. Now, imagine there are tools that exist that we can cut and paste these genes that allow these organisms to survive in this environment, or to survive in these environments, and paste together, and I'm simplifying this in many ways, paste together an organism that is able to do the best parts of A and the best parts of B, right? What can we do then if we take these newly engineered strains and then experimentally evolve them and see just how well they're improved? This is not, no kind of crazy talk or whatever else. There's a whole field that's dedicated to that. So understanding the tools or developing tools that we can apply to engineering these extremophiles for this purpose. Now, I'm gonna close here soon, actually, but people always ask, like, why do this anyways? Does it really matter? Well, it's obviously that, we are approaching a dawn of age where space flight is going to be possible. Now, the amount of dollars that is required to, send a payload up to space is astronomical. It's very, very costly. But if we could take microbes and engineer them such that they're able to mine different planetary bodies or produce different things that we like, we now become independent in space, or our future descendants become independent in space by being provided with food, renewable energy, and also, like I just showed you, some of these raw materials like plastics that can be used and collected right there and then used for our purpose. So that's kind of like the thoughts that I have with respect to kind of like where science is going and microbes and what at least interests me. And I'll end right there. But first, I'd like to acknowledge kind of my graduate advisor, Rich Walensky, my postdoctoral advisor at University of Idaho, Christopher Marks, collaborators, Rohan Madamsediai, he's at Duke. He worked with me on the metagenomic sequence data for the anaerobic work. Tomislav Tisak, who also worked with me on the PhD project, particularly in engineering the ability to utilize these aromatic compounds in the Cordae Siriki, as well as an undergrad, Joe Warren. And with that, I could take any questions.
All right, so good question for you. You mentioned, many, many generations, 50,000 generations, I think at one point, of tardigrades, which I think begs the question, how long does a tardigrade live? And Alex specifically asks, how do you know that you've gone enough sort of generations deep in doing your experiments on evolution?
So with respect to the tardigrades, the 50,000 generations I was talking about, actually E. coli, the Lenski long-term evolution experiment, I was working with that. In terms of the lifespan of tardigrades, they're very, very hardy organisms. I'm not sure what their lifespan is per se. Again, they can dry out, become, essentially desiccates, right, for 15, 20 years. And once water and nutrients are present back in their environments, they revive and wake up. It's called cryptobiosis, right? So I'm not sure their lifespan per se. So that was the first question. The second question is, evolution is open-ended, right? So you could design an experiment and say, hey, I want to have an experiment, done for, 120 generations. Or in the case of Rich, for example, the LTE now is at about 75,000 generations, right? What is interesting is when you're looking for, or when you're evolving these populations, you want to set up your experiments such that you're always looking at them, at intermediate stages, right? Maybe I test their growth rate. Maybe I look at their cell size and shape. Maybe I sequence them. Maybe I look at transcriptomics, right? You do these things in between your experiment and you will find what changes are occurring. And then you decide whether or not you want to extend your experiment, right? They're relatively low cost. They don't take up much space typically or much resource. So how do you know when you're, an evolution experiment is enough? Whenever you say it's enough. That's what my answer would be.
Wow. So I'm going to combine two questions. Courtney and Angela basically are wondering about the application of this to maybe predicting or understanding variants in the evolution of viruses. And I think COVID and the flu immediately come to mind. But would you say there are ways that this applies?
Yeah, so I know there are some groups who have conducted evolution experiments, not much so with with SARS-CoV-2. So this is the pathogen that actually causes COVID-19. But you don't want to evolve a super virus, right? But with other viruses with similar features, right? And then looked at the evolution. So yes, it is a way for you to look at variances that may crop up given a certain set of conditions, whether or not you're in a lung cell, for example, or a kidney cell or whatever else. So yes, you can use that to study changes, right? Or predict changes that may accrue with evolution in a certain environment. Absolutely.
So Jeff and Anna are both asking about space travel, and in particular, we'll say microbes, but Anna also asked about tardigrades. If you're sending tardigrades to the moon, as I guess an Israeli team did, according to the question here, or just for the bioethics of space travel in general, like, are there ethical considerations when you're sending these microbes around space?
I mean, so there is absolutely bioethical considerations. I tend to tell people when I hear this or when I think about this, it's kind of like, so at this moment, we don't know if there's anything that exists there. So typically the arguments are, okay, if you introduce something, what does that mean for the natural fauna? And we don't have to worry about that, right? Because we have at least no evidence that those exist. Again, and then these are microbes. So one can then argue and say, well, these are agents that we're sending, right? I mean, are we scared that they're going to be alone? Is that what the argument becomes then? I'm not sure. If we're talking about humans and, going to those places, I think the ethics, right, makes more sense there, right? Because, there are limitations. You have a human that's being subjected to high doses of radiation, crazy weather, isolation and all of these things. So I think the ethics apply more there to them and not to microbes, at least from my perspective. And what are we contaminating? I think that, we're adding things to a planet with the hopes that, or to a moon or, whatever, with the hopes that we can mine or collect resources and provide a space for us to go or enable us to go to these places later on with reduced costs to us. So I think it's okay. That's how I'll answer that.
So Susan wants to know, so if you can find a microbe that can survive radiation, is there any application to using that as some sort of like a protective coating for other life forms?
I think that is possible. I think that it would require you to have a lot of microbes on the body or on this life form. I just use it more generally speaking. I think in terms of kind of like protection from radiation, I think to use natural structures, right? Like caves or lava tubes that are found on Mars and some of these other places, I think that's more ideal. However, I will say that roaches, cockroaches, they've been around for a very, very long time because of, their ability to block radiation like that as well too, those hardy things. So yeah, I mean, it's a strategy that could work. Whether or not it would scale to humans, I doubt.
So can you give some other, Prem is asking this question, some other examples of plants where microbes help plants survive in tough situations?
Where microbes in tough, I mean, I think we can just normal like beans plants, even like, I don't think it's a tough situation, but when we think about the symbioses and root nodules with nitrogen fixation and microbes providing them nitrogen, right? Without that, it would be tough for them, right? Harsh is a relative term. At least that's one. There's also kind of in hydrothermal vent systems, there are microbes associated with like, and this is different, I don't want to call them plants, but like tube worm type things, right? Where the microbes provide resources, elemental resources that allow these plants to grow. And that's another example, I'll say.
Jackie wants to know, I think, Jackie, this is your question, are the microbes sort of shielding against radiation, or can they actually eat radioactive waste? And is anything like that being developed or in use currently?
I mean, so there are microbes that do eat radioactive waste. In the case of Dinococcus radiodurans, for example, it's been a long time since I looked them up, but how they deal with radiation is that they have DNA repair mechanisms that are able to repair the mutations, the damage that's caused to DNA because of the radiation. So they just work extremely fast to repair, mismatches and things of that nature. So they do a very, very good job of that as well. There are other organisms as well that deal with radiation because of pigments, so melanization. I mean, fungus, they do stuff like that. Fungi, they do stuff like that. So those are three different examples of how radiation is dealt with across the spectrum of life.
George is wondering if you have, I'm going to broaden it out. He says a text reference for astrobiology. So I'm going to assume George is a pretty advanced reader on the subject. And then I'll also say just a general, more pop sci book on astrobiology you might recommend.
Pop sci, I mean, so typically, I like to read Nick Lane. He had a couple of things. It wasn't really necessarily, I won't say astrobiology related, but I tend to think about, his text in that way. That's fine. In terms of papers, there are a bunch of papers that just recently came out. So typically on astrobiology, you have to really search for astrobiology journals. But in more microbes, there's one that just came out actually this week. It was published in eLife. I got it yesterday in an email, and I read it. It's someplace. But yes, I can provide that to George's.
Okay, we can also try to do a link when we put it up or something like that.
Perfect. Yeah.
So Alex actually has an interesting question. Like, at what level of, now can you, if you can, I don't know if your lab is like this, but if you have an understanding of like the levels of biosecurity one uses in a lab setting, what level security is yours? And can you just sort of go over how complicated things can get with like recirculating the air and trying to keep people safe?
I see. So, I mean, again, so like I said earlier on, only about 1,400 microbes can cause disease to humans. So typically, what the issue is kind of like the reagents and the things that you're working with in terms of these studies. Like, so radiation, you need to take special, courses or classes, right? Radiation courses, do environmental health service practices, etc. I myself, currently, I'm working in a BSL-2 lab, but that's just because I'm working with a pathogenic organism that causes cholera, right? So that's what I'm working with. I'm not doing anything related to astrobiology at all right now. But again, it's just something that I think about. And when I worked previously with E. coli, right? So E. coli, there are some strains that cause disease and some that do not. Ours was non-pathogenic. That was a BSL-1 lab. So I don't think that these organisms, given the fact that they live in these extreme environments, you may have to go and collect some things too. That can be extreme, right? And a hazard as well too. For example, the geothermal field in Ethiopia, right? 122 degrees Fahrenheit. That's hot, right? So there are some considerations to make there, but in terms of bringing things to the lab and being able to work with them, I think the main issue becomes the resources and the things that you're using to grow them.
So I think you may even have opened your talk. I know you've talked about this a couple of times in our prep call and stuff about the idea that you were born to be a microbiologist. Can you elaborate on that a little more? Like, what was it that just like drew you into the field? Like, what's great about it?
I see. So I won't say necessarily I was born to be a microbiologist. I didn't know, but I would say that in 5th grade, I just definitely remember this. So in 5th grade was the first time that I had an experience with a microscope. It was this one monocular thing, and Miss Shingles was the teacher's name at Mosaic Academy of Saginaw. And she brought in a microscope, and we made these slides. We collected water. And I just looked in the microscope, and I was just in so much awe, like that there was these living things that I did not see, I didn't know about. Yes, we had heard about germs, but there was like no kind of ever like, oh, this is what we're talking about when we're talking about germs. Again, not all of them are germs. And just seeing things just like swim around, attacking each other and everything, like a little city in this drop of water, it was just so fascinating for me. It just made me so exciting. And my trajectory, however, and before that, I used to collect bugs too. So I was, if I was to say I was born to be a biologist, okay, yes, that's probably what it was. That's something I've always been interested in, like just like life in general, plants, trees, whatever else. But I kind of got off track in my trajectory in life. And it was in high school, actually, that my science teacher, Mr. Painter, he brought in a video. And the video basically asked a question. Here was the premise. Given what we know about life on Earth, if we change the parameters, let's say we change the amount of the wavelengths of light that's coming from the sun, let's change the density of air, let's change gravity, and life evolved on these planets, how would it look, right? That was the whole thing, like a whole hour of all of these different phenotypes that could emerge. And that absolutely sold me in terms of kind of thinking like, hey, when I'm thinking about microbes, I already had kind of like an elementary understanding that microbes live in crazy places. I'm thinking to myself, well, space is, harsh and hostile environment. If they can live in these environments here, then obviously they could live there, right? So just by understanding them. So really at that point, I became blown away. Now it's kind of tough to talk about those things, in a field or in a place where it's kind of like, oh, we're interested in microbes that cause disease. We're interested in microbes that are going to, make money somehow, right? It's kind of, tough because if you want to start a lab, if you want to be successful in science, you need funding, right? So you need to kind of study your research in that type of way all of the time to kind of like maximize your funding. But those things were absolutely always interesting to me in that way. Yeah.
Well, it sounds like you really got on your path because of 2 great teachers.
Absolutely. Absolutely. Just doing something that, was, for where I grew up and outside the norm, right? Absolutely.
So as you talk about sort of reimagining the world, I think this ties loosely into Jeffrey's question here. A variety of plant and animal species are threatened with extinction. So what is the effect this extinctions or mass extinctions may have on different microbial communities?
Yeah, absolutely. Although I say, like I said, microbes, they are very, very, they've been here forever, right? They've been here, from the dawn of time, just quickly right after. We really can't put a date on it just because of they don't leave fossils, right, per se. But absolutely, I do think that the diversity of microbes, will decrease somewhat. How much, I really can't put a number to that. I haven't seen any studies that actually talk about that, like how much we're losing microbial diversity. I will say, however, that if, we change things like diet, if we change things like, again, we talked about this earlier, like the region that we're in, we can shift community composition, right, in our guts or in our host. So I think that as, climate continues to change, as humans start to infringe on different places, right, we're going to have, definitely going to have microbial compositional changes writ large. that's what I would say to answer that.
Elizabeth wants to know if having a community of microbes as opposed to a single organism can affect the way they mutate and evolve.
Yeah, so absolutely. So when we think about microbes, they're always competing for some, even single by themselves, a single population, they're always competing for some resource, right? They're always competing to inhabit a niche. So for instance, you have, you can have E. coli that grows extremely fast, and at the same time, you're producing acid that's going to inhibit another organism that's going to cause it to release some kind of system that's going to pierce its neighbor, et cetera, right? So because you have all of this fighting, this infighting, with each other, right? You're going to have different traits evolve, right? So for example, if you have bacteria and viruses that infect bacteria called phage, right? You can see that over time, you have these fluctuating dynamics where the bacteria says, oh, you're infecting me. I get a mutation that's going to cause you to stop infecting me. And the phage is like, oh, we have nothing there anymore. So here's a mutation. And again, these are random mutations that evolve. That's going to learn to infect you through another route, right? So you have all of these dynamics going on all of the time in multi-trophic systems, I think that's the right word there, that you wouldn't necessarily have, at a single cell, a single species or a single population level. Yeah.
You make it sound like a college frat.
I would say that's what it is.
So Jessica and our very own Sachi are asking about other aspects of your research where it might inform future pharmaceutical or therapeutic interventions, or even other applications like in food or plastics. Are there more examples sort of extrapolating from the last slide?
I see. Yeah, so currently I would say that I'm just beginning. So I just started my this position here at Michigan State University, where I am working with Vibio Cholera. So this is the pathogen that causes cholera. It infects about 1.3 million people per year, kills about, 2,000 or so, or 140,000 or something like that a year as well. And what I'm working on is kind of mapping all of the interactions. So you have a slew of mutations. So I'm mapping all of the interactions that causes a reduction in fitness in Vibio leading up to death, right? And the idea is that if we can understand these mutations that, it makes me grow slower and makes me stop growing, et cetera, we can use the genetic sequence information that we get from these organisms and then treat natural, we can use those ideas to treat these infections in the wild. So that's kind of like one thing. In terms of kind of like the other work that I've done, so again, with the Methylobacterium work that I showed you all, and again, so I didn't mention this earlier in the part of the talk. So Methylobacterium is the one that produced the plastic that I experimentally evolved to do so. Species belonging to that genus are actually the one that actually hitchhike rides to space. So Methylorurum, Methylobacterium, the family Methylobacteriaceae, those are the ones that actually go to space. So they're good in that regard. So they're already space ready. So in terms of that type of work, yes, I mentioned the plastic, but again, we're planning on cutting those genes out. At least my collaborator Christopher Marks, and introducing butanol. So now you have natural fuel production as well too. But microbes, like I said, they've been making medicines. They make vitamins. We use them for that purpose already. So yeah.
Harvey has a fun question. He says astrobiology tells us that microbes can inhabit other planetary bodies and travel through space. He wants to know when they do a study like the Lichen study or something like it, are they able to tell after 18 months, or is their equipment sufficiently sensitive to know if that material's been contaminated potentially by a non-Earth-based microbe?
So that would suggest, so we don't know if there's non-Earth-based microbes, right? But I would absolutely say we know. So, for instance, we take an organism, we put it in space. We know what we put, right? We have information about it before we put it there. Now, if we go and put it there and something comes back with that, right? Yes, there are ways where we can, deep sequence populations, and we map those things back to the sequences of the ancestor or the strain that we sent there, right? So actually say, okay, we sent ATGC CCA, do we get ATGC CCA back, right? Or something thereof. Well, of course, we'll get mutations, but if we get something totally different. And yes, so typically here on Earth, we measure, we read things such as the ribosome, these 18S ribosomes, 16S ribosomes, for example, in the case of bacteria, that actually gives us an identity of the bacterium that we're working with, right? So we always have a way that we can map and say, okay, we map this 16S and assuming that this non-Earth-based organism has the same genetic program, and we should be able to pick up stuff like that too, right? So yeah, absolutely, there are ways I would say to pick up that information.
I think I have time for 2 questions. You may limited time, but we're gonna try to do 2. Susan wants to know if we are underestimating the dangers of E. coli generally, and particularly in the elderly.
In the elderly, again, so as I said, E. coli, there are non-pathogenic forms and there are pathogenic forms, right? I think in elderly and, if you get an E. coli infection, particularly the elderly and young infants, right, it can be particularly deadly, right? Because they produce these toxins that cause your kidneys to shut down, right, and widespread sepsis. So I don't think that we've underestimated that as a field. I think these are things that we know. But again, not all E. coli cause disease, so keep that in mind.
Okay, and Frem is wondering, can using microbes potentially reduce methane in the environment and combat climate change? Are there applications for that?
Yeah, absolutely. So methane obviously is a gas, right? So it's going to, evaporate and things of that nature. However, I'm just going to, one quick little story I liked about 2 years ago now, maybe last year. So an organism found, a psychrophile found in ice in Antarctica. They're deeply embedded in this ice, right? They have no access to the outside world. But they have this very, very unique way of getting energy actually just from the air as it's passing by, right? They start collecting it there. So if we could think about, we find out how these organisms are doing that, perhaps we can make some non-specificity to, these gases and say, instead of pulling down, CO2 or water from the air, you pull down methane. Perhaps. I'm not sure, but, microbes have found a way to pull things down. I'm sure that we can engineer them to pull down other things too.
So I'm going to invite in. There she is. Okay, I didn't even have to invite her. You're right.
I just showed up. You didn't even have to invite me.
Welcome to the party.
And thank you so much for that, Nkrumah. I couldn't necessarily figure out from your website all of the things you do, but that's why we invited you onto our stage, so you could just talk to us about it. And thank you.
Yeah, you're welcome. Thank you. I'm still figuring out what it is that I do.
Well, we thank our audience as well for being here, and we hope you will come back in about a month's time when we will issue another invitation when we have somebody who will talk to us about cute aggression, which again, I had never heard of that. I thought, well, that's something we might all be interested in. So tune in about a month from now, and you too will know all about cute aggression.
I just can't wait.
Yeah, exactly.
So I'd like to thank Courtney Sloan, Sashi Gerben, Jeff Bitchman, and Ameche Ukpali for technically producing this event. Thank you guys all, and we'll be back. What was the date again, Ann?
About a month from now.
About a month from now. All right, thanks. We'll see you soon. And some of you will be joining us in the VIP Q&A right now. We'll see you there.